Hello students, welcome to KENE'S Educare, a group of KENE solutions. This is part 9 of lesson The Fundamental Unit of Life. So as you all know that in this part we were studying mostly about the cell, and in the last class we have even started studying about uh, the cell organelles, right? We have discussed about cytoplasm, we discussed about the endoplasmic reticulum. Even we discussed on that there are two types of endoplasmic reticulum. One is smooth endoplasmic reticulum and the other one is rough endoplasmic reticulum, right? So those are the two different types of endoplasmic reticulum that we discussed on in detail in the last part, where we have even discussed on cell organelles, what exactly they are, what is the need, and what is the function that they perform in detail, right? So today, for now, I want each one of you to please revise each and everything that we have studied so far in the lesson. Like whatever notes you have taken in your book, please read that, everyone. I hope everyone is revising each and everything, every notes that you have taken in your book, as that is very important. Okay, so everyone please go through it quickly. You have to go through each and everything that you have written, each and every diagram that you have drawn, everything. Okay. So like after you're done with this, I will be even like showing you all the slides one by one as we were doing it before. Okay. So we started this lesson by discussing about the cell theory. So like Robert Hooke, he was the first person, like as he was studying his like doing his activity on cork, which is a substance that comes from bark of a tree, right? So he found out there's something like there's some structure which resembles a honeycomb like structure which has some little compartments in it, right? So this was in the year 1665 when Hooke made this chance observation through a self-designed microscope, and Robert Hooke called it as Box's cell. Okay, so I want you all to please go through it once about the cell theory. So after this cell theory, we discussed on like what exactly is a cell, and as we just now discussed that cell was first discovered by Robert Hooke, and this was in the year 1665. Now cell is derived from a Latin word which means little room or a compartment. So now this is one of the historic, you can say, statement which has been loved by all, that cell is the basic structural and functional unit of life, and this cell has certain organelles present in them which perform a specific function. So we are studying about the cell organelles right now, right? We studied one of the cell organelle, that is endoplasmic reticulum. Now we have vacuole, nucleus, plastid, and if you remember in the last class, we even discussed on the difference between the plant cell and the animal cell, and along with that we even discussed on like the similarities as well. Not only the difference, we even discussed the similarities, right? Then after this we discussed on like what are living organisms made up of. So we discussed that through an activity 5.1, right? So I just want each one of you to please go through it quickly. After this we discussed on like what exactly while you do the experiment for onion peel, what is the exact observation that will be visible to you. So like for that you need to go through the slide and I want each one of you to please go through it. After this we discussed on like this activity 5.2. So I just want you all to please go through it. After this we discussed on this different shapes and sizes of the cell, right? So I just want you all to please go through all of it. Then we discussed on like what is a cell made up of and what is the structural organization of a cell, right? I hope everyone is going through plasma membrane, which is also called as cell membrane. Now, this is again a very important part. Like every component of the cell are very important. Okay. After this, we discussed more about diffusion and osmosis that takes place, which is again very important. So please have a quick glance through the slide as well, as this slide is again very important. Okay. I hope everyone is going through this slide because on this, like after this slide, we discussed on three types of solution, that is hypotonic, hypertonic, and isotonic. So these are the three different types of solutions which you should be like knowing till now. We are just doing a revision now, so make sure you know that as well. This is what I was talking about. So we discussed on hypotonic, hypertonic and isotonic solution, right? So everyone please go through it quickly. Then even like you need to draw this three different types of solutions, like as you have just read about it, you know about it. You need to show when the cell will shrink and when the cell will shrink and when it will absorb water and will increase in size. Okay. Then we discussed on this activity 5.3 with osmosis, how it exactly takes place with an egg. So here we have the protocol. You should be knowing the observation and what is the result, or you can say like conclusion for it as well. Okay. After this, we even discussed on this activity where we were discussing about the dried raisins and the apricots and how exactly osmosis and diffusion takes place over here, right? So here we have the process of endocytosis which has been explained, and I want you all to please go through that process because endocytosis is a process which mostly takes place in amoeba, right? Then we discussed on cell wall. So always remember that plants only have cell wall, animals do not have it. Okay. Then we discussed on this activity as well, so I just want you all to please go through it. I hope everyone is like knowing the procedure. You can even like come up with the observation and the conclusion for the same, right? After this, like this is the proper, you can see the procedure that you need to know, and this is how like the activity has been done, right? After this, we discussed on nucleus as well, right? I hope everyone, like even like while you write about nucleus, you have to draw the diagram that has been shown on screen, showing the nuclear envelope including the outer membrane and the inner nuclear membrane. Then they have nuclear pore, chromatin, nucleolus, ribosomes, rough endoplasmic reticulum. Everything needs to be shown. Okay. So here we have more information about nucleus, because nucleus is one of the most important part of the cell, as it includes the DNA. Now DNA stands for deoxyribonucleic acid. So that DNA, it has all the information that we get from our parents, right? Then here we have like a note on prokaryotic and eukaryotic cells. I just want you all to please go through it. Then we discussed about cytoplasm in detail. So you have to go through the diagram as well as the information given. Then after this cytoplasm, we discussed on like about the viruses, because viruses are as such that they do not like they cannot stay outside the body, human body, so they need a body for the existence and for the survival and in order to like multiply as well. Okay, so please go through this slide as well. After this we started discussing about the cell organelles, where the first organelle that we had discussed was endoplasmic reticulum, right? But before we move on to that, I want you all to please go through this slide because here we have some of the information on cell. So please go through it, everyone. After the cell organelles, now here we have some of the names which have been given, or some of the cell organelles which have been known, and I hope every one of you have like gone through that as well. So after this we discussed on the endoplasmic reticulum, right? So I want you all to please go through it. I hope everyone is going through this endoplasmic reticulum, and I hope every one of you know that there are two types of endoplasmic reticulum: smooth endoplasmic reticulum and rough endoplasmic reticulum, right? After this, we discussed on like the differences and the similarities that exist between the plant cell and the animal cell, right? So the structure has been given and we have discussed some of the points. So I want every one of you to please go through it. I hope everyone is like going through it. Then now, today we'll be discussing about Golgi apparatus. So as you can see the structure, how exactly Golgi apparatus looks like, right? So Golgi apparatus has lumen, it has cisternae. Now cisternae is one of the most important thing that like you should label every time you draw Golgi apparatus. Okay. Then there's an incoming transport vessel and an outgoing transport vesicle. Okay, you should know this. Incoming transport vesicle and outgoing transport vesicle. And there are some newly formed vesicles which are even being shown, right? So see, Golgi apparatus, it was first described by Camillo Golgi, which consists of a system of membrane-bound vesicle that is called as flattened sacs, which are arranged approximately parallel to each other in stacks called cisterns. Can you see, like the membrane bound organelle, which we know it as flattened sacs, how they are arranged parallelly to each other in stacks? So those stacks we know it as cisterns, and these membranes, they often have connections with the membranes of ER. So the membrane that you can see over here, they have some kind of connection with the membrane of endoplasmic reticulum, and therefore it constitutes another portion of a complex cellular membrane system. Okay, what does it forms? It forms a complex cellular membrane system because of the connection that it has with the membranes of endoplasmic reticulum. Okay, so the material synthesized near the endoplasmic reticulum is packed and dispatched to various targets inside and outside the cell through the Golgi apparatus. So whatever material that is going to get synthesized near the endoplasmic reticulum, it is going to be packed, and after packing it will be dispatched to other targets, like whatever targets are there. After packing it, it will be dispatched to different targets inside and outside the cell through this help of Golgi apparatus. So one of the function of Golgi apparatus is transport the synthesized material, like whatever material is you can say synthesized near the ER, it is going to collect it and it is going to transport it inside and outside the cell with the help of Golgi apparatus. So its function includes storage. It is going to store, modify and pack the products in the vesicle. What it is going to do, it is going to its function includes storage, modification, and packing of products in vesicle. So in some cases, complex sugars may be made from simpler sugars in the Golgi apparatus. So this will happen, not in all the cases. Okay, what will happen? Now when it comes to the function of Golgi apparatus, it includes storage, modification, packing of product in the vesicle, and in some cases what will happen, even the complex sugar will get modified, or like complex sugars will be made from the simple sugars in the Golgi apparatus. So the Golgi apparatus is also involved in the formation of lysosomes. Where is this Golgi apparatus involved? It is involved in the formation of lysosomes. Now we will be studying about lysosomes as well, and you will come to know that lysosomes are called as suicide bags of the cell. And I'm just like telling you to have that information, so that you know what is the function of lysosome, and you know this thing because you should be knowing that which cell organelle is called as the suicide bag. So it is the lysosome itself. Okay. Now we have some information on like the person who has discovered Golgi. So his name is Camillo Golgi, and based on his name, the you can say the component of the cell was been named. So Camillo Golgi, he was born at Cortino near Brescia in year 1843, where he studied medicine at the University of Pavia, and after graduating in 1865, he he continued to work at Pavia at the Hospital of St. Matteo, and at the time, most of his investigations were concerned with the nervous system. What was his investigations concerned with? It was concerned with the nervous system, and in 1872, he accepted the post of Chief Medical Officer (CMO) at the hospital for the chronically sick at Abiote Grasso, and he first started his investigation into nervous system in a little kitchen of this hospital, which had converted into a lab. So just look at the hard work that he had done. Okay, and the journey he started with. So, like while he was doing his investigations on nervous system in the kitchen of the hospital, the work of greatest importance which Golgi carried out was a revolutionary method of staining individual nerve and cell structure. And this method we now know it as black reaction. So while he was doing his investigations on nervous system, he came up with a certain you can say procedure which is now known as black reaction, where we are able to like stain the nerve cells and even the cell structure. So this method of black reaction, it uses a weak solution of silver nitrate and is particularly valuable in tracing the processes and most delicate ramifications of the cell. Okay. What this black reaction involves? It has a weak solution of silver nitrate, and uh, that silver nitrate is valuable in tracing the processes and most delicate ramifications of the cell. So all through his life, he continued to work on these lines, modifying and improving this technique, and Golgi received the sorry, highest honors and awards in recognition of his work, and he shared the Nobel Prize in 1906 with Santiago Ramon Cajal for their work on the structure of nervous system. Okay, so it's just a brief discussion on Camillo Golgi, what are the work that he has done, and how exactly he was working on Golgi apparatus and the nervous system. So before proceeding with lysosomes, I want you all to write about Golgi and even draw the structure of Golgi in your book. I hope everyone is writing it. After this, now we have to study about lysosomes. So see, structurally, lysosomes are membrane-bound sacs which are filled with digestive enzymes. What are they filled with? They are filled with digestive enzymes. And these digestive enzymes, they are made by RER. Now, what is the full form of RER, everyone? It's rough endoplasmic reticulum. Okay? What is the lysosome made up of? Digestive enzyme, and these digestive enzymes are made up of RER, that is rough endoplasmic reticulum. So lysosomes are a kind of waste disposal system of the cell. What are lysosomes? They are kind of waste disposal system of the cell, and this help to keep the cell clean by digesting any foreign material as well as worn out cell organelles. Okay, so the enzymes that are being made by RER, that is rough endoplasmic reticulum, they help the cell to remain clean. How that happens? By digesting all the foreign materials as well as whatever worn out cell organelles are present. Okay, that has been removed. So foreign materials entering the cell such as bacteria or food as well as old organelles, they end up in the lysosomes which break complex substances into simpler substances. What happens over here? Whatever foreign materials are going to enter the cell, like as in case of bacteria, food, then there are certain old organelles which have been present, right? So they are all going to end up in the lysosomes, and that is going to break the complex substance into simpler substance. Now lysosomes are able to do it because lysosomes have very powerful digestive enzymes, and this powerful digestive enzymes, they are capable of breaking down all organic material. Okay, what are they capable of? They are capable of breaking down all organic material. So during the disturbances in cellular metabolism, for example, just imagine or suppose that the cell gets damaged, then in that case what lysosome is going to do is lysosome will burst and the enzyme is going to digest their own cell. What will happen? Lysosome will burst and the enzyme is going to digest their own cell, and because the enzyme is digesting their own cell, that is the reason we call this lysosomes as the suicide bags of a cell. Okay, now if you look at the anatomy of the lysosome that has been shown, so it has a plasma membrane which has a lipid bilayer, then we have hydrolytic enzyme mixture, and along with the hydrolytic enzyme mixture it has glycosylated membrane transport protein. What does it have? Glycosylated membrane transport proteins which have been present. Okay, so you should be clear, like why are lysosomes known as the suicide bag of the cell? Because lysosomes are going to burst and whatever enzymes are present in the lysosome, they are going to digest their own cell. So that is one of the reason why we call lysosomes as the suicide bag of the cell. Okay. I hope everyone is writing all the important points from the screen. Okay. So I hope everyone has like taken a note of all the points with respect to lysosomes, whatever has been given on screen. Now after this lysosomes, in the next class we will be studying about mitochondria in detail. Then we have plastids to discuss, vacuoles, and then cell division. So mostly in two more parts we will be completing with the lesson. Now I'm stating two more parts because we have summary and some of the exercise questions to discuss as well. So in one class, in one part, if I'm completing mitochondria, plastid and vacuole, the other class I can start with cell division, because see, the information on cell division is only given for two paragraph, right? Mitosis and meiosis, but it doesn't get completed over here. Okay, we have many things to know about cell division, so we'll be discussing it in bit detail. Okay, so we'll study about cell division, and after this type cell division, that is mitosis and meiosis, we will be doing the summary revision in short, what exactly we have studied in the whole lesson. Okay, then we have some of the exercise questions to discuss over here, and I want each one of you to please go through this exercise questions quickly. I hope everyone is going through the questions given on slide. I hope everyone is going through the question that I'm showing you on screen. So this is how we will be ending up with the lesson, the fundamental unit of life. But I hope like whatever we discussed today about the lesson, the fundamental unit of life, you all are very much clear with it. So now the rest of the part for the lesson I'll be continuing in the next part. Thank you everyone.